Hi Year 12s. Today we're going to be looking at cell control, which is focusing on the cell cycle and the different stages of the cell cycle. We'll very briefly look at prokaryotic cell cycle, and then we're going to move on to eukaryotic cell cycles, including the factors, internal and external, that can affect cell division and regulate cell division. So let's begin. As per usual, you've got your need to know slides.
What have I learned?
So these are all the SACE requirements that you need to know for this topic. Not going to give you time to write them down on this video, so make sure you pause the video if you want to write them down and tick them off as we go through the different points. The cell cycle. So what the cell cycle actually is, it's a series of events that occur inside of a cell before that cell divides into two daughter cells. It occurs in both prokaryotic cells and eukaryotic cells. This slide's focusing on prokaryotic cells, so the cell cycle in bacteria. And the cell cycle in bacteria occurs in three different stages: B, C, and D. Stage B is the time between the end of cell division and the beginning of DNA replication. So we can see B here. Technically it would start over here. So at the end of one cell division, right up to the point where DNA is about to replicate. Then we start the C period, which is the time between when DNA is duplicated or replicated, and between when cell division actually begins. So C period is just when our single circular chromosome, is going to be replicated. And then we have the D period, which is the time between the completion of DNA replication, so straight after DNA replication has taken place, and the end of cell division. And at that point, we'll start back into B. Okay, so very, very simple cell cycle for bacteria, a simple type of cell. In eukaryotes, though, it's a little bit more complex. So the cell cycle, again, will occur in three different periods, which is the interphase, mitosis, and cytokinesis. Interphase is by far the longest of the three stages, and it's actually broken up into three stages itself: G1, S, and G2. During this time, the cell goes through a whole lot of metabolic reactions, such as DNA replication and protein synthesis, helping the cell prepare for cell division. There'll also be a lot of growth, as you'd expect. You then go through mitosis, which is the stages of prophase to telophase. During these stages, the nucleus of the parent cell is broken down and the chromosomes are split, and we create two identical daughter nuclei. And lastly, we have the third stage, cytokinesis, the physical division of the parent cell into two genetically identical daughter cells. So we've already looked at mitosis and cytokinesis in our cell division PowerPoint. So this PowerPoint is focusing purely on interphase. We're going to look in depth at the three stages of interphase: G1, S, and G2. Over here we can see the entire cell cycle. Mitosis is here, and cytokinesis actually happens in this part as well. The rest of it, the blue, green, and yellow, that's all interphase. Okay, so this is by far the longest part of the cell cycle, and you can see that a majority of a cell's life is actually spent in interphase, and it's a very short time spent in mitosis. Looking at interphase in a little bit more detail, as I said before, it is the longest part of the cell cycle, and during this entire phase, the cell is going to go through a series of metabolic reactions, helping the cell prepare for cell division. So, the three stages of interphase: G1, S, and G2. The G1 phase lasts for roughly around 10 hours, and during this phase the cell begins to grow. We're going to have metabolic reactions such as protein synthesis occurring, building up the proteins that we need for the rest of our cell's cycle. The cell is also going to start to collect and gather up and accumulate free DNA nucleotides and ATP in order to prepare for DNA replication, which is going to happen in the S phase. So the S phase occurs for around 9 hours. During this time, DNA replication takes part on every chromosome in the nucleus, and we end up with 2 copies of every chromosome attached by a centromere. The centrioles are also going to divide in preparation for cell division, so that we have 2. Lastly, we go through the G2 phase, which lasts for about 4 hours. Some of the organelles in the cell are going to be replicated, so that we've got organelles for both copies of the daughter cells. ATP is going to be synthesized to replace any lost ATP used in G1 and S phases, and the cytoskeleton begins to be pulled apart to help assist in mitosis. And lastly, protein synthesis occurs. So they are the three stages of interphase. We're looking at roughly 23 hours roundabout for interphase. clearly the longest part in a cell's life cycle. The important part about talking about the cell cycle, though, is that there's checkpoints, and these checkpoints are checking that the cell is in the right progress or right stage of progress in order to move on to the next step in the cell cycle. The cell cycle can be stopped at any of those checkpoints, and there are 3 checkpoints. We've got the G1 checkpoint here at the end of the G1 phase, we've got the G2 checkpoint at the end of the G2 phase, and we've got the M checkpoint during the mitotic phase. So each of these checkpoints are looking for their own unique different things, and like I said, at any point during the cell cycle and any one of these checkpoints, the cell cycle can be stopped to allow more time in a specific step. So the G1 checkpoint at the end of the G1 phase, what's happening here is the chromosomes are checked to ensure there's no damage, so no mutations or anything like that has actually occurred to the chromosomes. They're all good to be passed on to a daughter cell. The amount of free DNA nucleotides, the amount of DNA polymerase and enzyme, and the amount of ATP is all assessed. If any of those 3 aren't at the optimal levels, so we don't have enough of those points, or if there's mutations that have happened in the chromosomes, the cell will physically not enter the S phase. It'll just stop there and wait. Now, obviously, if the reason that the cell didn't enter S phase was because the DNA nucleotides or DNA polymerase and ATP weren't in good enough amounts, the cell can wait in the G1 phase until those amounts are at the optimal conditions. If it's in case of a mutation or damage to a chromosome, obviously it's going to be very hard to move on to that S phase because the chromosomes have damage that we don't want passed on to the daughter cells. The G2 checkpoint, so we're moving on from the G1 checkpoint, G2, all the way over here. So we've gone through the S phase, and at the end of the G2 phase, we're going to end up with the G2 checkpoint. During this phase, all chromosomes are checked to ensure that they're replicated, make sure that they've all been copied over. Again, each chromosome is going to be checked for mutations to make sure no mutations have happened in the DNA replication stage. And if something has gone wrong and there is a mutation, the cell will not enter mitosis, so as that mutation is not passed on to a daughter cell. In the mitotic phase, we go through the final checkpoint, the M phase, which is happening just here, where sister chromosomes are going to be reviewed to make sure they're actually joined by the centromere, and to make sure that that centromere is actually attached to both spindle fibers of the cell and can be pulled apart equally. They are the 3 checkpoints that the cell goes through in the cell cycle. Now, it's not just the cell cycle that controls cell division, it's also the external factors. So cell division can actually be controlled by internal and external factors. Internal factors are obviously factors that are occurring inside of the cell's environment. External factors are going to be factors in the extracellular environment. External factors can actually be chemical or physical factors. We'll look at physical factors first. So there's 2 different physical factors that we're going to look at. Density dependence and anchorage dependence. Density dependence is basically a phenomenon where animal cells stop dividing once a single layer of cells has been formed. And more cell division won't actually occur unless one of those cells in that layer dies or is removed. So you get one layer of cells, cell division stops. One of those cells dies or is removed from the layer, then cell division will occur again to actually replace that and ensure that that layer of cells is full. Anchorage dependence, on the other hand, is when animal cells will stop dividing if they're not physically attached to the extracellular matrix or some other physical surface. Chemical external factors, on the other hand, could be something like growth factors. So growth factors will normally bind to receptor proteins that are embedded in the cell membrane, and when growth factors actually attach to that receptor protein, it begins a metabolic pathway inside the cell which stimulates growth. A good example of a growth factor is hormones, and one specific hormone is the human growth hormone, HGH, which is secreted by the pituitary gland in the brain. Once that hormone's been secreted, it travels through the blood into the target cells, which are generally found in the muscles and the liver, where it binds to those receptors and initiates cell division. As it initiates cell division, that's going to cause growth. HGH is actually responsible for dwarfism and gigantism, so obviously low levels of HGH will mean there's less cell division happening and less growth happening, which will be dwarfism, and high levels of HGH means more cell division, more growth, which will be linked to gigantism. Internal factors. So we've looked at the external factors, at physical and chemical, but there's also internal factors. So internal factors are factors that are found inside of a cell that are going to help control cell division. Most of these are going to be cyclins and CDKs. So the cell cycle is actually regulated by cyclins, which is a type of protein, regulatory protein, and also by cyclin-dependent kinases, which is a type of enzyme which we're going to refer to as CDKs. The main purpose of cyclins and CDKs is they actually control those cell cycle checkpoints that we looked about in a few slides ago. When they're activated, CDKs, their purpose of them is to transfer a phosphate group to a target molecule. When they attach that phosphate group to the molecule, it signals the cell to move on to the next stage of the cell cycle. CDKs have to rely on cyclins, and cyclins will bond with CDKs, forming something called a cyclin-CDK complex. Once that complex has been formed, again, it will stimulate the cell to enter the next phase of the cell cycle. Eventually, cyclins will dissociate from CDKs, which prevents a cell from entering the next stage. So, the cyclins and CDKs and the other proteins that help regulate the cell cycle are actually all created from genes called proto-oncogenes and tumor-suppressing genes. Any mutations in these genes can obviously cause cancer because it's going to alter what cyclins and CDKs actually do. So cancer. Cancer is a disease caused by unregulated cell division. Nothing new there. Unregulated cell division can result in the formation of something called a primary tumour. Now, a primary tumour can break off, but it's generally in one spot. It's stuck in one location. It can break off and move to other parts of the body in a process called metastasis. When a tumour has broken off and started to spread to other parts of the body, it's now the new tumours created because of it are known as a secondary tumour. Mutations that occur in the proto-oncogenes and tumor-suppressing genes are a problem because they can actually cause unregulated cell division. If they cause unregulated cell division, it's going to result in the growth of a primary tumour. That primary tumour can obviously break off and then move around the body and create secondary tumours. Mutations that happen in those genes, so proto-oncogenes and tumor-suppressing genes, are caused by something we call carcinogens. A carcinogen is any chemical or any agent, like a virus, that can influence the development of cancer.
That's all, folks.